In this series of videos, we're going to be learning about a family of organic molecules called proteins, which occur in every variety of life that exists. Now, before we can talk about protein structure, we need to talk about the foundational building block from which all proteins are constructed, which is this molecule here, which is what we call an amino acid. Now, people with a strong chemistry background will recognize why amino acids are given this name, because if we look at the left side of this molecule, we can see an organic nitrogen functional group is what we call an amine, which we know is going to act as a base when it is put into water, while on the right side of this molecule, we can see that there is a COOH, meaning that we have an organic acid, also known as a carboxylic acid functional group here, hence the term amino acids. The final component of amino acid structure is what we call an R group, which is actually not a single structure. The R group is actually a variable group, meaning that there are a number of different chemical structures that can take the place of the R that we see here. Now, throughout this series of videos, you're going to be hearing the term peptide a lot. Peptide is simply the scientific word for anything that relates to proteins. So, in general, whenever you hear the word peptide, there is some sort of component of a protein that is going to be discussed. For example, because an amino acid is the foundational building block, and because we're only looking at one amino acid here, this is what we call a monopeptide, meaning a singular peptide. Now, if we wanted to take multiple monopeptides and join them together in order to make a protein, we would need to do a chemical reaction that involves reacting the amine that exists on one side. So here we can say that the amine is giving up one of the two hydrogens and reacting with the acid on the other amino acid adjacent to it. So here we can see the acid functional group is giving up its OH group in the acid, and when we take OH and H and join them together, we get water. So the joining of two monopeptides involves the removal of water, and in the resulting whole that we create, a new bond forms connecting the two monopeptides, and because this is a bond between two monopeptides, it is not surprisingly called a peptide bond. And remember that peptide is just a word referencing anything related to proteins, so a peptide bond is the type of bond that holds all protein structures together. Now, not surprisingly, if a single amino acid is called a monopeptide, two amino acids joined together in this reaction are what we call a dipeptide. And if we were to do this reaction again, so if we were to take a third amino acid and take the amine group and react it with the acid group at the end here, we would have three monopeptides joined together, which not surprisingly would be what we call a tripeptide. Now, if we continue this reaction indefinitely, hundreds, thousands, or even hundreds of thousands of times, we would end up with a structure that we call a polypeptide, meaning we have many monopeptides that are joined together, and when these are folded into specific structures that each have a very specific function within an organism, we can now call this type of structure a protein. But the actual process of forming a protein is something that we'll look at in subsequent videos. Now, one important skill that needs to be done, number one, is reading a polypeptide structure, and number two, being able to identify how many different monopeptides are joined together. And there's a strategy that we can use, and the first of which involves looking at the symmetry or lack thereof of a polypeptide. So here we can see that the left side and the right side of this molecule do not look the same, and therefore we can say that polypeptides actually have a front side and a back side. So if we look on the left side, we can see that there is an amine group here, and we've written this tripeptide structure in this order for a very specific reason. So the end of a polypeptide structure where you find an amine is what we call the N-terminus. So terminus is just a fancy word that means end. The verb to terminate is simply a fancy way of saying to end or to stop. So the end of a protein that has an amine at the end is what we call the N-terminus. So that would be on the left-hand side of this molecule. If we go all the way to the end, we can see that we have an acid functional group at the other end, and because carbon is the central atom of the acid functional group, we refer to this end as the C-terminus. So in this case, the C-terminus of this tripeptide would be on the right-hand side. Now, if you want to identify how many monopeptides join together in order to make this, all that we need to do is look at the number of acids and amines that have been joined together. So if we look at the central structure, we can see nitrogen, carbon, carbon. This carbon here used to be the acid, and this nitrogen here used to be the amine, and right here we have our first peptide bond. Then if we go NCC, here this double bonded oxygen used to be an acid group, and this nitrogen used to be an amine group, and there is our second peptide bond, NCC, and then we get to the C-terminus. So the number of NCC repeats that we have will tell you how many amino acids are actually joined together. Now, the reason why this tripeptide is written with the N-terminus on the left and the C-terminus on the right is we read polypeptide structures the same way that we read English, from left to right, and we always start with the N-terminus and end with the C-terminus because this is actually how cells produce a protein. They start with the amino acid at the N-terminus and add, extending the C-terminus toward the right, like this. Now, the final thing that we need to discuss are the R groups that make up the last component of amino acids. And R groups, rather informally, are what we call functional groups, and I'm putting this in quotes because chemistry students will have a slightly different definition of functional groups, but let me explain what we mean by this when we're talking about R groups. Now, because the R group can actually be a number of different structures depending on which amino acid we're looking at, the specific R group that an amino acid has can affect the shape of the protein that they're part of. For example, we can see a very small R group may be a single hydrogen atom, whereas a very large R group can be this bicyclic ring structure that we see here, and obviously this is going to change the shape of what the final protein is going to look like. But more importantly, different R groups do not always have the same polarity. For example, if we look at all of these R groups, we can see that they are almost entirely composed of carbon and hydrogen, which makes these particular R groups non-polar, which is going to affect their behavior when the protein that contains these amino acids is dropped into water. So because all of these R groups are non-polar, we would expect that they would be hydrophobic and will naturally avoid water and stay close to each other, attracting to each other using their dispersion forces. However, if we take a look at these amino acids here, we can see that pretty much all of these amino acids contain a strongly electronegative atom such as oxygen or nitrogen, which means that if they are dropped into water, water is therefore going to be able to hydrogen bond with the dipoles that we find in these R groups. So because we can draw dipoles here, these R groups are obviously polar, which means because they can form hydrogen bonds with water, we would expect that these would be hydrophilic and want to be close to water to form hydrogen bonds. Now, the reason why we spent so much time reviewing how acid and amine functional groups react when they're dropped into water in terms of how they ionize, this is the main reason why. So if we look at the ends of each of these R groups here, we can see that they used to be COOH functional groups, meaning that they had an acidic R group, which means that in water they undergo an ionization by losing their hydrogen, forming H3O plus in water. But what is left of the COOH group, functional group is negatively charged because, again, water pulled off the hydrogen, leaving them with a negative charge. Now, naturally, all negative charge is going to want to attract to some sort of positive charge, so this is radically going to change the behavior of how these amino acids will move inside of water. Now, conversely, these particular R groups all have amine functional groups, and we know that amines function as a base when they are dropped into water because the lone pair on nitrogen is going to pull a hydrogen off of water, making OH minus, which gives the nitrogen on each of these amine functional groups a positive charge. So because all of these R groups are positively charged, they will want to seek out some sort of negative charge to form an attraction. In the next video, we're going to use our understanding of R groups in order to predict how a polypeptide is going to go from a simple straight chain like this and fold itself and form more complicated structures like the one that we see here.